So let's begin this discussion of primary structure and let's kind of walk ourselves in order of what's typically done in a laboratory. So in the lab, I'm basically given a protein, and this protein I'm told that I'm going to have to figure out the primary sequence of. Well, before I can take anything away from the protein structure, I'm going to have to destroy the protein that's given. So that's one of the things that are very done in the very first step of these analysis is to look at your protein, figure out how to destroy it. And there's a couple of ways that we do that, almost in order. And one is you want to destroy the disulfide bonds, the disulfide bridges. That's very important. We talked about the purpose of cysteine and cysteine making these disulfide bridges, these linkers in the protein that kind of make it hold its shape somewhat. So the very first thing that we have to do is we have to get rid of those. And we do that by using a reagent. And the reagent here is 2-mercaptoethanol. And once we use 2-mercaptoethanol, we basically destroy those disulfide bonds and we convert them back to SH bonds. So this is kind of how they would show up in the first place. Okay, so after we destroy the disulfide bonds, the protein begins to unravel. Okay, so once the protein begins to unravel, we now probably don't have any kind of glob system in place. We get something a little bit more linear, something a little bit looser, and something that really glue isn't holding together anymore. But this is one of the things that we have to do in order to destroy all of that. I also want to mention that there's other type of bonding that can hold these proteins together. Disulfide bridges are not the only ones. You get all kinds of van der Waals forces that begin to hold them together as well. So you start to see these hydrogen bonds exist. You start to see hydrophobic areas and hydrophilic areas inside of the protein. We'll take a look at that when we go to tertiary and quaternary structure. But for now, we're only going to be focused on the disulfide bridges because those have to be destroyed. That really is a bond that I have to make sure is taken care of before we go any further. So once the disulfide bonds are broken, it's now my job to go in and figure out what amino acids are making this protein up. And the very next thing that I typically do with these proteins is I put it into an acidic environment. And typically this is nothing more than just 6 molar hydrochloric acid. That's all that it is. And then I also need to put some heat onto it. So this is at 100 degrees, so this is the boiling point of water, and I hold it at that high acid, high temperature for at least 24 hours, so a very long period of time. And what begins to happen is that it takes this protein and it begins to chop it up, and it will cleave all of the peptide bonds. So that's the purpose of step number 2. This cleaves the peptide bonds. Okay, and what we get as an end product are individual amino acids. The next part of this whole system, number 3, is that I then 10 would probably send it through an amino acid analyzer. Now we've talked about the amino acid analyzer already in a previous video, and this will tell me the exact amino acids that make the protein up, and it will also give me the number of each. So it gives me an idea of how much alanine is there, how much glycine is there, how much tryptophan is there. It will tell me that arginine was not found. It will go through the protein and it will literally basically analyze every single amino acid and give me the actual content of all of it. So very important for number 3. That's the power of the amino acid analyzer. Again, remember, the amino acid analyzer typically works off of ninhydrin. So this is something that you want to be careful with. Make sure your plumbing doesn't leak. Make sure that you don't turn everything purple and like a Smurf. So that's something that you kind of want to take priority with. Make sure that you're taking care with it. Make sure that instrument's up to par. So, those are the 3 steps that we typically take before we start analyzing any protein at all. It lets me get a very good idea of what amino acids I'm working with, how many of them, how complex is the protein, how large is the protein, how small is the protein, but that is the order of how to determine a primary structure. Now, there's a couple of other things that we can do as well, and one of these is how to determine the N-terminus of the protein itself. Right? This is our first player. So N-terminus is our very first amino acid. And it's very common for me to want to know what amino acid and I'm starting with, and it's a very easy reagent that I can slam into the picture. So what I would do is that I would add something called Edmonds Reagent. And Edmonds Reagent reacts with the N-terminal amino group, and it cleaves it, and it pulls it off, and then that piece can be analyzed from that point on. Now, again, we can go through the mechanism, we can go through the actual reaction curvy arrows. We're not going to do that, though. That's not the purpose of this class. I keep saying it over and over and over and over. The Edmonds reagent looks at the first amino acid in the chain and it cleaves it off. The Edmonds reagent, if you do want a name, maybe the name is kind of important, is called phenyl isothiocyanate. And this is basically a C double bond S, double bond N, and then off of this nitrogen is a benzene ring. So there's your phenyl, your isothio, there's your thio group, cyanate. You can kind of see that C triple bond N in a way that's involved. We abbreviate that as PITC. So that is the abbreviation for the big name, phenyl isothiocyanate, or Edmonds reagent typically is how it's referred to. So Edmonds reagent will take basically a protein and it will let me know the first amino acid in the actual chain. So let's say this is my protein that's been unraveled, and I add Edmonds reagent to it. And let's say that Edmonds reagent pops off and it says, well, okay, you've got the PTH is the abbreviation that we use for that. You've got this PTH alanine. That basically is letting me know that this is Edmonds reagent product, and it has pulled alanine off. So that means alanine is in the very first spot in the amino acid chain. So very, very important. Edmonds reagent, it's as simple as that. You add a reagent to it, you allow it to react, and then you identify it using chromatography with a standard. Edmonds reagent can't continuously be used, though. We can use this about 50 consecutive times. So what that means is that we can take the protein, add Edmonds, cleave it off. Take the same protein. After this one gets cleaved, we got a new first one. Add it, cleave the second one off. It will let me know what comes second. And then third, fourth, fifth, sixth, seventh, about 50 times we can use the Edmonds degradation product. However, this is basically due to how advanced your instrumentation is, because this is really automated. So this automated process, it's really dependent on the type of instrument that you're using. Some of them can actually go 100 or more times, but on average we're looking at 50 degradation products that we can get from this protein sequence based on Edmonds degradation. Okay, the problem is that all of these side products begin to accumulate in the actual protein solution, and that begins to interfere with the products. So it's these side products that begin to kind of take over the protein after a certain point, and then you're going to have to kind of clean up and do something else with your protein other than Edmonds reagent. So that's the story with Edmonds reagent. That's your first reagent that we can put in to get an idea of what amino acid is coming first in a main chain. So in the next video, we're going to, if we can have a reagent that's looking at the first amino acid, then we should be able to see a reagent that looks at our second amino acid. And that is what we'll focus on in the upcoming video. So stay tuned. Another 10 minutes of your life is not going to be wasted. We'll take a look at what looks at my tail end, the caboose of the amino acid chain.